Good morning and good evening. I am Shormishta De, professor in Department of Biophysics and Indian PI of LASI-DAD project. I welcome you all for this webinar. The LASI-DAD is the Harmonized Diagnostic Assessment of Dementia for the Longitudinal Aging Study in India. It is a depth study of late life cognition and dementia. This study recruited LASI respondent aged 60 and older from 22 states and union territories across the country. LASI, that is a longitudinal aging study in India, is a nationally representative survey of the health, economic status, and social well-being of the Indian population aged 45 and older. LASI-DAD has been conducted in two waves so far. Wave 1 from 2017 to 2020 and Wave 2 from 2022 to 2025. It is one of the family of population-based cognitive impairment and dementia studies using the Harmonized Cognitive Assessment Protocol, which includes the health and the retirement study and its sister dataset around the world. The LASI-DAD Webinar Series started on May 8, 2025 and has so far discussed topics such as cognitive changes, caregiving, and biomarker studies, and many other things. So today, Dr. Masroor Anwar, a scientist of LASI-DAD project, will present on the association of neurodegenerative blood-based biomarker with cognitive outcome. The biomarkers are p-tau, NFL, GFAP, and the ratio of A-beta 40 and 42. Following this, there will be a discussion on the correlation among the neurodegenerative biomarkers in India and the United States to harmonize the cognitive assessment. So Masroor, you can start now.
Yes, thank you, ma'am, for the introduction. So, good morning, everyone in US, and good evening to all audience in Asia and Pacific. So, I am Dr. Masroor Anwar, working as scientist in LASI-DAD project at All India Institute of Medical Sciences, New Delhi. So, today I'll be speaking on the topic blood neurodegenerative biomarkers in dementia. And by the end of this session, you will have a clear understanding of how biomarker research has evolved and why it is so important to have a biomarker research like LASI-DAD or HRS, the data that I will be comparing with later on in my presentation. More importantly, I will be showing the data of LASI-DAD work that we have done and compare those data with the HRS study, which has been done in US, and how there is a correlation between these two data. So I'll start with a brief. So before starting that, here are the four terms which needs a brief discussion. So first, I will be discussing about the biomarker. Second is the blood-based, why biomarker should be blood-based and why it is important to have blood-based biomarker. Third is neurodegeneration, which is very important, especially when we age, and what are the neurodegenerative diseases. And finally, the dementia, which is age-associated neurodegenerative disease. So starting up with biomarkers, so NIH in 2001, the NIH working group actually came out with a definition, standard definition of biomarker, what it is. So they defined biomarker as a characteristic that is objectively measured and evaluated as an indicator of normal biological process or pathogenic processes or pharmacological response to therapeutic intervention. Later on, a study review came in 2022 by Eric et al. They showed, here we can see like biomarker, why it is important in case of disease also and for treatment also. Here you can see biomarker is important for diagnostic purposes, for diagnosing having a diagnosis of disease. And also biomarker identify the risk of disease. If we have a specific biomarker, we can predict the risk of developing that disease. Third is having the prognosis of the disease. Now, in case of treatment, what happens? Biomarker exactly, it can predict the treatment. If we know early, also it can monitor the treatment. And biomarker can also monitor the treatment response, and how a treatment is safe. Biomarker can also tell you that. So it is so important to have a biomarker. Now, looking at the historical prospect, so if we look back in history and see that earliest human in early human history around 300 BC, it was Herophilus for the first time, an ancient Greek physician who came up with indicator of disease and reported that body temperature is one of them, and also the pulse rate. These early in early times they were used as an indicator of disease. In modern era, we can see here, like in 1776, after a long time, because of the advancement of the research and the technologies that we have today. So in 1776, the first biochemical marker was identified and reported by Matthew Dobson. It was glucose for diabetes. And first molecular marker that we can see here, it was reported in 1847 by Henry Benz Jones, who for the first time reported a protein marker as for the disease multiple myeloma. So since then, uh, uh, people have been working around looking at the biomarker. Now, why blood-based biomarker is important in health sciences? So first, we know that any sample that we are taking from the patient or it has to be like minimally invasive. So blood-based biomarker is minimally invasive and easy to obtain, and it is simple, fast, and safe. Compare with if we compare it with the tissue biopsies or collecting the CSF samples. Now, the second most important thing is if we have a blood-based biomarker, we can detect it early, and that detection helps in treating the patient and have a better treatment outcomes for any disease. Third important thing is real-time monitoring of the disease. If we have, we can take multiple time. If it is required, we can take repeated blood samples and can track the patient disease progression. So real-time monitoring of the disease can be done. And fourth is like cost effective. The blood-based biomarker are comparatively cheaper, and it has got high throughput, and more importantly, it is suitable for large studies like LASI-DAD or any large-scale studies. Now, fifth important point currently these days is in precision medicine. Blood-based biomarker helps, like say for example, the genomic marker, the diseases which involves mutation in the genes, and there has to be a developer precise if we have to have a precise medicine for a particular patient or particular individual. So blood-based biomarker will be very helpful. Now, how the publication has come up in the field of biomarker, if we start from 1873, when for the first time biomarker term was reported in the publication. So how it has progressed, you can see that in 2000 till 2010, there was very few studies. Like in 2000, total biomarker study in all the diseases was around 2000, which had a steep increase. By the time we are reaching in 2025, almost fourfold increase are there in the publications coming up in biomarker. This was reported by Jeffrey Aronson in 2019. Now, when we specifically look for neurodegenerative biomarker, we can see again by 2000 until 2020, almost only 1,000 publications were there in total related to neurodegenerative biomarker. And and there's a reason for it. Because of we have a there is advancement in cellular and molecular biology, and the technology that we have these days, it was not available previously. So it is because of this reason we see that there is more number of publication coming up in biomarker field. So now if we specifically look into the neurodegenerative biomarker, which is blood based, so again, some 100 publications were there around till 2020, which sharply increased almost to 3 to 4 fold. You can see almost 500 publications has come up and till now, like in 2025. So these are the publications that are coming up in biomarker research. So the next topic is neurodegeneration that I will be dealing with. So neurodegeneration, as per definition, it is a progressive loss or damage of nerve cells in the brain or the peripheral nervous system, leading to gradual decline in neuronal function. And it has got the certain symptoms we can see if at all there is neurodegeneration. First is like memory loss or the confusion or motor problems. This may be influenced by genetics or the environment or because of aging. Most important thing about neurodegenerative disease are it is incurable and treatment only involve management symptoms. Now, if we see the archival note, the neurodegeneration was noted for the first time by Jean Charcot in 1800, mid 1800, where he speculated that it is because of neurodegeneration, the nerves is degenerating, and it is because of these things there is a characteristic loss of neurons in humans. So why neurons are vulnerable? So there are specific reasons for that. So neurons, they are long-lived and they are rarely replaced. So this is one of the reason neurons are vulnerable, and the neurons have got high metabolic rate, and it is which leads to more oxidative stress. And we know that neurons are highly interconnected, and if there is dysfunction, it spreads across all the network, which leads to finally neuronal death. Here we can see the cascade of events which leads to neurodegeneration. So what happens first, as we age, the mitochondrial decline occurs, leading to DNA damage, which further leads to impairment in the protein clearance, followed by synaptic loss, and which causes the inflammation in the neurons. And because of oxidative stress, vascular defect starts to occur, which causes the neuronal death. If we look at the mechanism of neuronal degeneration, a healthy neurons is there first. Now, what happens because of genetic damage, the DNA gets impaired or mutated, and which leads to the diseases like Huntington or Friedreich ataxia. These are because of genetic mutations or the repeated sequence. Now, if there is a protein misfolding or there is a mutation in the protein, so it is overexpressed and forms a plaque or tangles in the brain, which further leads to neurodegeneration. Or in case of microglia, which has got, if there is a damage in the signaling process, so it leads to inflammation and causes neurodegeneration. Inflammation itself causes neurodegeneration directly through the synaptic signal loss, which causes the neuron death. Now, what are the major neurodegenerative diseases? It includes Alzheimer disease, Parkinson, or amyotrophic lateral sclerosis, or frontal temporal dementia, Huntington disease, or spinocerebellar ataxia. All these diseases, if we see that it is localized uh, due to the atrophy in the specific region of the brain. If we see if there is some atrophy in the basal ganglia region of the brain, so it will develop diseases like Parkinson disease, Huntington disease, Alzheimer disease, or FTD, frontal temporal dementia. If there's a problem in thalamus, then FTD, AD, or PD can occur. If there is a problem in hippocampal region, the AD, we can see AD in those patients. If there's a problem in cerebellum region, then spinocerebellar ataxia will occur. Similarly, if there is a problem in the brain stem region, then ALS or SCA can occur. Now, talking specifically about brain atrophy, because of amyloid plaque formation or neurofibrillary tangent formation. So we can see here severity of disease. If we compare the stage 1 with a stage C, then we can see that in frontal cortex and olfactory regions, the A beta plaque formation is higher, it's very severe in these regions, which leads to AD. And about NFT, neurofibrillary tangle formation, which primarily occurs if we compare the stage 1 and 2 with the stage 4 and 5, then we can see particularly it occurs in olfactory bulb region, hypothalamus, temporal lobe, and parietal lobe. So these regions of the brains are affected. Now here we can see like pathology of Alzheimer disease. This is healthy neuron section, brain section from healthy uh, sections of the brain. So neurons are intact. And in case of AD, this is a section from AD patient, brain sections from AD patient, and we can see here neurofibrillary tangle formation is there, along with amyloid beta plaque deposition. Primarily it occurs because of these four reasons, and inflammation is most important of them, and damage to which causes damage to the neurons. Now, why globally it is very important to have dementia research? So we can see here, specifically we are concerned about the rising number of dementia cases in the world. If we see the dementia cases in 2030 in Americas, it is approximately 15.8 million, which will just double around 2050, approximately 30 million cases will be there of dementia. Now in Asia, if we compare, you can, because of the large population, Asia has got the highest number of cases, like 67.2 million cases will be there in 2000. It is expected to have like 2000 by 2050, which is expected to be 38.5 by 2030. So because of these reasons, we should have studies which should look into dementia research. Now I'm sharing here like a few very important study going on around the world, longitudinal studies. The major international research consortia like working on dementia research. Cosmic is one of them. Cosmic is one of the largest longitudinal study, which includes almost 40 cohorts across 6 continents over which includes almost 20 countries and with 200,000 participants. What they are doing exactly, Cosmic study aims to pool harmonized data to understand the determinants of cognitive aging. The other study which is going on is Worldwide Fingers. It is a global network of clinical trial of dementia prevention. The other study is Global Burden of Disease study, which is being carried out to check the Alzheimer's disease and other dementia, focusing on incidence and prevalence and disability adjusted life years. Now, the DIAN study, it is being done in North Europe, Asia, Europe, and South America. Some of the regional study that is going on, North America is doing this study, ADNI and HRS, which we are associated with. One very interesting thing we can note here that one of the report is there which says that North America is the one of the largest dementia treatment market for so it is almost expected to spend almost $6.2 million by 2023. So major study which is going on in Europe are like CFAS, TELDA, ELSA, and SHARE study, which is a joint study between European nations and Israel. And why Europe is more concerned about dementia, like it has got 2020, if we compare the data of 2021 with 2050, we can see here that almost per 100 inhabitants, there are 23 affected people with dementia, which will just double by 2050. For example, in Italy, then Spain and Denmark, the least cases are there in Britain. Now, the study which is being carried out in Asia and Australia are these are the studies like and the country involved in these studies are South Korea, Japan, China, and Australia. And here in India, we are doing LASI-DAD study, which is a longitudinal study of diagnostic assessment of dementia. And one of the study which has been reported by Benish et al. In 2024, it said that East Asia has got the highest prevalence, like almost 70% cases are there of dementia in East Asia, and Southeast Asia being the least, almost 7 to 9%. About LASI-DAD, briefly, I'll say that LASI-DAD is the first and the only nationally represented study on late life cognition and dementia in India. It includes almost 4,000 samples and which includes almost 22 states of the country of the population of the participants over the age of 60 years. And if we look at the distribution and the prevalence of the disease, Jammu and Kashmir and Odisha is more prevalent. The cases are more here, and prevalence in West Bengal and Assam is comparatively less. The other green regions are having the least number of cases. So while LASI-DAD study is important, especially in India, now we can see that dementia in India, the prevalence in 60 years and above is approximately 7.4%, and almost 8.8 million Indians over the age of 60 lives with dementia. Upon analysis, it has been shown that dementia is more prevalent among females compared to males, and rural population is more affected with dementia compared to the urban population. Now, these are the parameters which has been included in LASI-DAD study, like memory, learning, attention, executive function, language fluency. Geriatric assessment includes anthropometry, height, weight, arm and calf circumference, knee height, blood pressure, including ADL and AIDL. Now we are more concerned about the biomarker study, which includes blood samples that we have collected, serum, which includes separation of serum, plasma, and Buffy code. LASI-DAD study is also taking up the genomic data and are doing the next generation sequence to find there's variation in the genes. It is also taking the data for taking the MRI data for neuroimaging data is being collected to which will enable to have a brain volumetric study analysis. It also includes the socio-demographic data. So now I will be showing the data of comparative analysis of neurodegenerative biomarker, blood-based biomarker, and will compare the LASI-DAD data with HRS. Now, what the brief methodology of this comparison? These two studies are largely nationally representative data of LASI-DAD and HRS, which has got in HRS almost 300. The total participants are 3,466 participants are there, which has got all the four markers. And for LASI-DAD, it is 2,151 participants with all the four biomarkers available. So these two, the data from these two participants will be compared. Now, the platform that we have used is Simoa SDX, which is one of the most advanced and reliable platform to analyze the data, which was validated across the two countries, like HRS study, which was done at University of Minnesota, and the LASI-DAD, which was done at AIIMS, New Delhi. Now, these are the molecules that we have considered for our biomarker study. That is amyloid beta 42 and 40, tau protein, NFL, and GFAP. Now, why these molecules are important? All these molecules have been found to be associated with neuronal network and signaling, and there are ample research which has been previously done to show that it is associated with dementia. Now, how this molecule comes into blood is important thing, because blood-brain barrier doesn't allow these molecules to move into the bloodstream. So A beta primarily binds to LRP1, and through the secreted LRP1, it moves through the transcytosis, it moves into the blood circulation. Similarly, tau, because neurons are like it gets the blood-brain barrier actually through transcytosis, it moves into the bloodstream, and this through transcytosis it moves into the bloodstream. GFAP protein, it exactly when there is a damage to the astrocyte, GFAP protein crosses the blood-brain barrier and enters into the bloodstream. Similarly, NFL, which through exosomes, it enters the blood-brain barrier. Now, for comparison, 81 samples were taken and cross-calibration was done both at Ames and University of Minnesota. The correlation coefficient came to be very good, around 0.99, thereby showing the platform has got a little bit or no variation. So we moved up with a comparison of these two studies and found that when we look at the distribution of neurodegenerative biomarker in India and United States, we can see here for the blue line here, it represents the HRS study data, and the orange line, the orange peak here shows the LASI-DAD data, distribution of LASI-DAD data. So if we see here, the US group shows the peak at lower GFAP value. Here you can see it is around 40 to 60, and for the LASI-DAD it is around 80 to 100. So on an average, we can say that GFAP level are higher in Indian cohort compared to the HRS. For NFL, both the country peak at low NFL values, and the HRS, the LASI-DAD curve has heavier towards the right. You can see here, meaning that more people with high NFL values are there in LASI-DAD compared to HRS. Now, upon distribution study of neurodegenerative biomarker between HRS and LASI-DAD, here we can see like A beta, the ratios of A beta, it is suggestive of if the ratio is lower, so likelihood of having Alzheimer's disease means having A beta burden. So here we can see the amyloid burden is, for both the cases, you can see the amyloid burden distribution is broadly similar for both the countries, but India tends to have slightly lower, you can see here, lower ratios compared to the HRS. And when we analyze the beta 1 at 1, here we can see that India shows longer values than here. You can see, so depicting that LASI-DAD cohort shows higher value of tau pathology compared to HRS. Now, comparing the correlation of neurodegenerative biomarker in India and United States, we can see here, if we compare LASI-DAD with HRS, we can see the correlation of GFAP with NFL. So the correlation here is 0.40, and for HRS it is 0.47. So if at all there is an increase in GFAP, corresponding increase can be observed for NFL also, and similar is the case for HRS. Now, if we compare the p-tau with GFAP, there is still a moderate correlation. 0.14 is for LASI-DAD and 0.28 is for HRS. So there is moderate correlation for beta also. For A beta, you can see here there is no positive correlation. No positive correlation. Thereby, it has got almost uh, no correlation for A beta compared to beta or NFL. Now, if we look at the level of protein here with the age, so these are the standardized neurodegenerative biomarker compared in different age groups, 60 to 70, 71 to 80, and 81 and above. You can see here that there is an increasing trend in both the study. Like for GFAP, if you see in LASI-DAD and HRS, in both the study, you can find a significantly higher concentration is there in both the study. Like 81 and above is having almost four-fold increase in GFAP concentration. Similar trend can be seen for NFL also. So with increasing age, the concentration of NFL increases in both the study. If you precisely look at the NFL level of LASI-DAD, it is higher compared to HRS. Now, looking at the A beta ratios of in LASI-DAD and HRS in different age group, so there is no difference in the level of A beta among the different age groups, either in LASI-DAD or HRS. Looking at the p-tau association, the level of p-tau 181 was significantly higher in old age in HRS, but it is not there in LASI-DAD. Here you can see. And now, looking at the cognitive score and its association with neurodegenerative biomarker in HRS and LASI-DAD, here we can see that for GFAP, the coefficient is you can see here for LASI-DAD, the coefficient is negative, thereby indicating that with higher GFAP, there is worst cognitive score, and same is the case in HRS. For NFL also, we can see both HRS and the LASI-DAD shows a negative coefficient correlation. So strong negative effect can be seen in HRS. For NFL, looking at A beta, you can see that it is almost there is no correlation. The coefficient is near zero or slightly positive. If we look at beta 181, the association is again negative for both HRS and LASI-DAD. So we can conclude from here like GFAP, NFL are all negatively associated with cognition in both HRS and LASI-DAD. If we look at the association of dementia with the biomarker in HRS and LASI-DAD, looking at their odd ratio in individual models and the combined models with biomarker factor score, then we can see that GFAP and NFL biomarker shows strong association. If you see that there is the odds ratio is towards the positive side for both the study. The odd ratio is almost same for both the study. So GFAP and NFL biomarker shows a strong association with dementia in HRS than LASI-DAD, except for A beta ratio. Now, if we analyze the association of cognitive decline with death, the neurodegenerative biomarker in LASI-DAD and the HRS, you can see NFL strongly predicts the death in LASI-DAD. You can see here the odd ratio is towards higher positive side, but in HRS, it is not there. So, association, the NFL strongly predicts the death in LASI-DAD, but it is not associated in HRS. So key findings of this study, NFL and GFAP shows higher level of association with poor cognitive function and increased likelihood of dementia in both HRS and LASI-DAD. Both markers predict cognitive decline in HRS but not in LASI-DAD. The A beta ratio, it is higher, in HRS, higher ratio were linked to worse cognitive function and greater dementia risk. In LASI-DAD, higher ratio were associated with better cognitive functioning. About PETA 181, it shows no association, showed no strong association with cognitive functioning in either population. And NFL and GFAP appears to be the most promising early indicator of cognitive decline and dementia. So regarding mortality, all biomarkers showed association with mortality, suggesting thereby they may reflect broader biological aging process. This is the publication which was done using these two data. The limitation of the studies, like there can be a variation because of the cross laboratory validation. Harmonization always helps, but doesn't eliminate the difference. Sometimes there are some confounding factors. And short follow-up period of about 4.5 years in LASI-DAD and 4 years in HRS may be a limiting factor. Future scope, the future research work towards improving the measurement and standardization of neurodegenerative biomarkers such as A beta 42 and 40 and PETA 181 were needed to ensure the reliability of cross-population comparison. Also, the longitudinal analysis, including upcoming biomarker waves from HRS and LASI-DAD, will be essential to evaluate their predictive values for cognitive decline and dementia. More importantly, expanding the biomarker panel to include more emerging molecules like p-tau 217 may further clarify the biological pathway linking to neurodegeneration to cognitive outcome. With this, I would like to thank you, and I would also like to thank the LASI-DAD team.
It's a very interesting talk. You have deeply discussed the neurodegeneration and the area of the brain which affected and the proteins that NFL, GFAB, A beta, tau, P tau, how they altered and damaged the neurons, and also the importance of blood-based biomarker and the data of longitudinal study going on in different parts of the world, and the publication, how it's increasing year-wise on neurodegeneration. And finally, the correlation of this plasma-based biomarker of Indian study and United States. So thank you, Dr. Mashroor. So now the session is open for question. If anyone have any question, the question is welcome now.
Okay.
I think no question is there.
Uh, doesn't look like there's any questions here.
One question is there, do you have vision
Uh,
impairment measures in your data set?
yeah, that we are right now
Not in
vision
this study.
impairment. Not right now, it is not there, but we are planning in our next wave.
Can you see the question, Mashur? Have you checked the
Yeah,
predictability through
ROC
ROC?
analysis. Yeah, ROC analysis has not been done uh, to predict the biomarker specificity, but the correlation study has been done, which I have shown in my slides.
It's a vast population study, so ROC.
Yeah, uh, like some 4,000 samples are there.
Okay, I think that might be it for questions.
Yeah,
But thank you so much, Meshwar, for presenting. Thank you everyone for coming. We will resume the webinars in the new year, in January with a presentation on the food frequency questionnaire and diet assessment in LASI-DAD. So please keep an eye out for the email notifications and on our website for when that will be announced. Thank you, everyone.
Thank you,
thank you.
Dr.
Thank
Masur.
you.
Dr. Day.
Thank you.
Thank
Bye-bye.
you.
Thank you.